Welcome to Between Sleep and Science, to a journey into the most profound library ever assembled, a realm where biology meets information science, and where the history of our planet is written in a language of elegant microscopic spirals. For eons, we have looked to the stars to find the origins of complexity, yet the most sophisticated technology in the known universe is already pulsing within you. It is tucked away in the quiet center of every single cell, operating with a level of precision that makes our most advanced supercomputers look like primitive toys. This is the world of DNA, the biological code that defines the living world. Imagine, if you will, the sheer, unimaginable density of this information. We live in an age of data. Huge server farms, massive hard drives, and cloud storage systems that span continents. Yet, if you were to take a single gram of DNA, a substance no heavier than a few grains of sugar, it could theoretically store more data than every piece of technology ever manufactured by the human race combined. If we could shrink the entire digital output of modern civilization, from every book ever written to every video ever filmed and every signal ever transmitted, it would fit into a tiny speck of genetic material. This is not science fiction. It is the reality of molecular efficiency. This incredible storage capacity exists because DNA doesn't rely on the bulky binary systems we created. Humans use zeros and ones, etched onto silicon or magnetic tape. Nature, however, uses a four-letter chemical alphabet arranged in a three-dimensional spiral. Unlike our digital devices, which require constant electricity, cooling, and hardware updates to keep data from degrading, DNA is a masterpiece of durability. When kept in stable, cool conditions, like the permafrost of the north or the dry silence of a cave, this biological archive can remain readable for hundreds of thousands of years, far outlasting any hard drive or optical disc. DNA is essentially a bridge. It is the bridge between the inert world of chemistry and the vibrant, pulsing world of memory and life. It is the microscopic machine behind your growth, the silent witness to your body's healing, and the guardian of your family's history. It flows through generations like an invisible river, carrying instructions that have been refined over billions of years of trial and error. It weaves forms from nothingness, builds structures of immense complexity, and holds mysteries that stretch back to the earliest dawn of biology. If these quiet wanderings bring you even a moment of calm, feel free to leave a few warm words, or tell us in the comments which city you're watching from, so we can see how far our explorations travel. You can also simply like and subscribe. Your support helps other weary hearts discover this cozy corner. And now, you don't need to do anything at all. Just settle in comfortably and let the slow, steady rhythm of deep time gently carry you away from your worries. Let the journey begin. When we think of history, we often imagine crumbling parchment, dusty library shelves, or weathered stone monuments. But there is a much older, more resilient form of record-keeping. Every strand of DNA is a living biography, a four-letter narrative that chronicles the deepest history any organism can possess. While the soft tissues of the body eventually decay, and even the strongest bones may crumble into dust, DNA possesses an almost supernatural ability to endure. It can survive in conditions where almost nothing else can last. Locked away in the freezing soil of the Arctic, the bone-dry silence of desert caves, or the lightless depths of the earth, where time itself seems to move more slowly. What gives this molecule its legendary toughness? It is a masterpiece of molecular architecture. The secret lies in the double helix, two long chains that twist gently around one another in a structural embrace. This spiral arrangement isn't just beautiful, it is a functional fortress. It makes the molecule strong enough to hold together through the violent process of cell division, yet flexible enough to bend and curl inside the unimaginably tight spaces of a cell's nucleus. The outside of the spiral is formed by repeating units that act like the sturdy rails of a ladder, shielding the precious chemical letters inside. This durability allows us to do something that once seemed impossible: communicate with the deep past. Scientists have become biological archaeologists, extracting fragments of genetic material from remains that are tens of thousands of years old. Even when heat, moisture, or natural radiation has begun to fray the edges of the code, certain stretches remain intact enough to be read. These lingering pieces are like whispers from a lost world. They reveal exactly what kind of plants once carpeted the earth during the Ice Age, how ancient animal populations migrated across vanished land bridges, and what environmental pressures shaped the ancestors of the creatures we see today. In the most extreme habitats on our planet, we see even more incredible examples of genetic resilience. There are microorganisms, the true survivors of Earth, that have evolved specialized repair systems. When their DNA is shattered by intense radiation or extreme desiccation, these biological engines move in to stitch the fragments back together with surgical precision. This ensures that their genetic message is carried forward, no matter how hostile the world becomes. Each surviving strand of DNA becomes a message in a bottle, cast into the ocean of time, waiting to be recovered and translated into a clearer picture of what once existed. By reading these sequences, researchers are uncovering ancient paths of inheritance that were once invisible. We are discovering how specific traits shifted over millennia, how organisms adapted to sudden climate shifts, and how the alphabet of life remained constant through it all. It is a world where chemistry is not just a reaction, but a durable archive, a bridge between the worlds of nature and technology that hints at a future where we may store our own libraries within the very chemistry that created us. While we often visualize DNA as a static image in a textbook, the reality is a breathtaking display of engineering in motion. The structure of DNA is arguably the most successful design in the history of the universe. It consists of two long, elegant chains that twist around one another to form the famous double helix. This isn't just a random coil, it is a masterpiece of molecular architecture where every atom has a specific purpose. This spiral arrangement provides a perfect balance. It is strong enough to maintain the integrity of our genetic information through billions of cell divisions, yet it remains flexible enough to bend, loop, and dance within the incredibly crowded environment of a cell. If you look closer at the interior of this spiral, you see a world of absolute order. Pairs of matching chemical symbols line up in a very specific sequence, creating a stable pattern that serves as a high-fidelity template. This design allows for a remarkable feat of biological duplication. When it is time for a cell to divide and create a new life, the spiral doesn't just copy itself blindly. It unzips down the middle, allowing each half to act as a guide for the creation of a perfect new partner. This ensures that the instructions for life are passed down with an accuracy that human engineering can only envy. The outside of this spiral is just as important as the code it protects. It is made of a backbone of sugars and phosphates that act like the sturdy rails of a ladder. This backbone provides the structural support needed to keep the delicate information inside from being crushed or tangled. Furthermore, the specific geometry of the helix creates grooves, narrow and wide paths along the molecule. These aren't accidental. They act as docking stations. They allow specialized proteins and enzymes to land at the exact spots they are needed to read a gene or repair a break, all without disturbing the rest of the molecule. But the double helix is just the beginning of the story. To fit inside the microscopic nucleus of a cell, the DNA must undergo a process of superpacking that would baffle any professional mover. If you were to stretch out the DNA from a single human cell, it would be about 2 meters long. To fit that into a space smaller than a grain of dust, the molecule wraps itself around tiny disc-shaped proteins called histons. Imagine these as microscopic spools. The DNA winds around them, creating a structure that looks like beads on a string. These beads then fold into thicker fibers, which loop and curl into even more complex shapes. This folding is not chaotic. It is a highly organized system of information management. Sections of the code that are not currently needed are tucked away into tight, inaccessible bundles. Meanwhile, the instructions the cell needs right now are kept in open, relaxed loops, ready to be read at a moment's notice. This dynamic architecture changes depending on the type of cell. A nerve cell will fold its DNA differently than a muscle cell, even though they contain the exact same instructions. By shaping itself in this intricate way, the molecule ensures that the right information is available at the precisely right moment, making the double helix not just a shape, but a living, breathing control system. If you were to look inside the nucleus of a living cell, you would find that the famous double helix is only the surface of a much deeper mystery. We often think of DNA as a simple, linear book, read from start to finish. But nature is far more efficient than any human author. The instructions within our genetic code are designed to be read in multiple, overlapping ways, a feat of information density that defies our traditional understanding of logic. A single region of the genome can actually carry more than one message, depending entirely on where the cell begins its reading process. Imagine a sentence that changes its entire meaning just by shifting your focus by one letter. In the world of DNA, this is a common occurrence. Sometimes, a slight shift in the starting point of the molecular reader creates an entirely different biological product. In other instances, the cell takes the initial transcript, a raw copy of the code, and physically rearranges the segments, snipping out certain pieces and stitching others together to create a variety of outcomes from a single sequence. This molecular editing allows life to produce a vast array of proteins and tools without needing an infinitely long strand of DNA. It is the ultimate form of space-saving engineering. Hidden within these long stretches of code are also secret signals that behave like the stage directions in a play. They don't necessarily build anything themselves, but they control exactly when and how a nearby region becomes active. Some sequences act as accelerators, speeding up the production of a vital enzyme, while others act as brakes, ensuring the cell doesn't overproduce a molecule that could become harmful. This multi-layered approach allows the genome to conserve an enormous amount of space, while supporting the staggering variety of functions required to keep a complex organism alive. Instead of assigning every single task to its own separate area, life weaves multiple layers of meaning into the same piece of code, creating an astonishing level of efficiency. To manage this complexity, the cell employs a team of specialized enzymes that act as a high-speed molecular assembly line. The process begins when a segment of the genome is copied into a temporary messenger form, a mobile script that can travel to the cell's manufacturing centers. There, coordinated components position each building block in the exact order dictated by the code. As this new chain emerges, it begins to fold almost instantly, twisting and curling into the precise three-dimensional shape required for its function. The accuracy of this system is nothing short of miraculous. These molecular machines work continuously, adjusting their pace based on the needs of the cell and responding to cues from the outside environment. They transform a silent chemical sequence into the diverse and active machinery that keeps life operating at every single moment. Whether it is a protein designed to carry oxygen through your blood, or a signal that tells a cell to repair itself after an injury, it all begins with this elegant overlapping script. It is a system that proves that in the world of biology, information is not just stored. It is dynamically lived, edited, and performed with a precision that bridges the gap between chemistry and art. In every second of your existence, your DNA is under siege. It is exposed to a constant barrage of environmental stresses, from ultraviolet radiation and chemical pollutants to the simple, everyday wear and tear of cellular metabolism. If our genetic code were a static blueprint, it would quickly degrade into a chaotic mess of errors, leading to biological collapse. However, the genome is not a passive victim of its environment. It is a self-monitoring, self-healing masterpiece. Within the microscopic world of the cell, there exists a specialized maintenance crew of enzymes that work with the speed and coordination of an elite emergency response team, ensuring that the integrity of our biological instructions remains intact. Cells are constantly patrolling their genetic material, scanning for the slightest physical abnormality. They look for misplaced units, unnatural bends, or chemical bumps that signal damage. When a flaw is detected, a sophisticated repair sequence begins. Imagine a team of molecular scissors that can recognize a damaged stretch of code with atomic precision. These specialized enzymes move in, pry apart the affected region of the double helix, and surgically remove the flawed section. But they don't leave a hole behind. Instead, another set of enzymes uses the undamaged partner strand as a template to rebuild the missing part, filling in the gap with perfect accuracy. Finally, a biological glue seals the backbone, making the molecule whole once again. This proofreading system is especially vital during the process of replication. Every time a cell divides, it must copy its entire genome, billions of letters of code. The potential for a typo is immense. To prevent this, DNA polymerases act like high-speed editors, checking each new symbol against the original as it is being laid down. If an incorrect letter slips through, the system pauses, backs up, and replaces it. Even when the damage is severe, such as a double-strand break, where both rails of the ladder are severed, the cell has backup strategies. It can either fuse the ends back together or borrow information from an identical neighboring chromosome to reconstruct the lost sequence. Without this continuous silent labor, life would be impossible. Our cells make rapid life or death decisions about how to fix every problem they encounter, keeping the genome stable over decades of life. This repair work is the reason we can survive exposure to the sun, recover from illnesses, and pass a functional legacy down to the next generation. It is a profound reminder that the story of life is not just about the information we carry, but about the incredible energy we expend to protect that information. We are built on a foundation of constant renewal, a living record of a system that refuses to break, no matter what the world throws at it. If you were to look at the genome as a book, you would see that it is never truly finished. It is a living document, a biological palimpsest, where new layers of information are constantly being written over the old. This is the engine of evolution, a relentless process of adjustment and refinement that has been unfolding for billions of years. While the repair systems we discussed work tirelessly to keep the code stable, they are not perfect. Occasionally, a tiny change, a mutation, slips through the cracks. These subtle alterations in the sequence are the raw material for the staggering diversity of life we see on Earth today. Most of these genetic shifts occur quietly during the massive undertaking of copying the genome for the next generation. Sometimes, a single chemical letter is swapped for another. Other times, entire segments of the code are duplicated, providing spare copies that evolution can experiment with. While many of these changes have no effect at all, and some are even harmful, every once in a while, a mutation creates a masterpiece. It might nudge a protein to work a little more efficiently, or shift the timing of a regulatory switch just enough to provide a survival advantage in a changing environment. When a change offers even a slight edge, it spreads through a population over generations, eventually becoming a defining trait of a species. But the rewriting of DNA is not limited to slow, random typos. Nature also uses a far more dynamic tool, the movement of genetic material within the genome itself. Scientists have discovered that some stretches of DNA behave like molecular travelers or jumping genes. Instead of staying anchored in one fixed position, these segments can physically leave their original site and settle into a completely new location. As they move, they can disrupt old instructions or activate nearby regions that had been silent for eons. They can even carry pieces of surrounding genetic material with them, creating entirely new combinations of information that could never have emerged through simple copying alone. This roaming behavior turns the genome into a dynamic, shifting landscape. It is a source of rapid innovation that helps life respond to sudden challenges. Over enormous spans of time, these traveling sequences have helped shape the very architecture of our cells, adding variety and fresh possibilities to the biological script. We see this play out in the history of every organism. Portions of the genome are duplicated, allowing one copy to continue its essential daily task, while the other is free to explore a new role. This gene duplication is one of the primary ways that complex new systems, like the mammalian eye or the avian wing, began their long journey into existence. All of these alterations turn our DNA into a living record of adaptation. It is a map of every victory our ancestors ever won against the forces of nature. Your genome contains the echoes of ancient relatives whose names have been lost to time, but whose survival strategies are still hard-coded into your very cells. It reveals that we are not fixed entities, but part of a continuous flowing experiment. The code is stable enough to preserve our humanity, yet flexible enough to support the emergence of new traits, ensuring that life remains a vibrant, ever-changing presence on this planet. When we look into the mirror, we see a human being. But if we look into our genetic code, we see a mosaic of the entire history of life on Earth, including some residents that might surprise you. One of the most startling discoveries in modern genomics is that we are not entirely human in the way we traditionally imagined. Scattered throughout your DNA are the remnants of ancient battles, a biological graveyard of viral infections that occurred millions of years ago. These are known as endogenous retroviruses, and they make up a staggering 8% of the human genome. To put that in perspective, that is far more than the total amount of DNA used to build all the proteins in your body. Long ago, in the deep reaches of our evolutionary past, certain viruses managed to insert their genetic material into the genomes of our ancestors' reproductive cells. Instead of causing a temporary sickness and fading away, these viral sequences became permanent residents, passed down from parent to offspring through countless generations like a family heirloom. Over eons, most of these viral fossils lost the ability to function as active infectious agents. They became broken, silenced, and buried under new layers of genetic code. Yet, their fragments remain, scattered across our chromosomes, as quiet markers of ancient pandemics that once swept through the prehistoric world. But these viral ghosts are not just passive baggage. In one of evolution's most brilliant moves, our bodies have actually domesticated some of these ancient intruders. In a process called exaptation, life has taken the raw material of a virus and put it to work for our own benefit. For instance, a protein essential for the development of the human placenta, the very organ that allows a mother to nourish her unborn child, actually originated from a viral gene. Without that ancient infection, the way we give birth and nurture our young might look entirely different. These viral remnants can also influence nearby human genes, acting as regulatory switches that help control our immune response or shape our brain's development. By studying these embedded sequences, scientists are able to peer into a window of the deep past. They can trace the outbreaks of viruses that vanished long before humans even existed, and learn how our ancestors' genomes fought back to neutralize these threats. These remnants are a record of survival, showing how life manages to turn a source of destruction into a source of innovation. It reveals that the genome is a master of recycling, capable of taking the most dangerous elements of nature and weaving them into the fabric of our survival. This viral legacy reminds us that we are part of a vast, interconnected web of biological history. We carry the ghosts of our enemies within us, transformed into silent partners in our ongoing evolution. It is a humbling thought that the very code that makes you, you, is a patchwork of ancient travelers, accidental hitchhikers, and repurposed invaders. Our DNA is a testament to the fact that life does not exist in a vacuum. It is shaped by every encounter, every struggle, and every tiny microscopic entity that has crossed our path since the beginning of time. If DNA is the musical score for the symphony of life, then your cells are the performers, and they don't always play the notes exactly as written. One of the most revolutionary discoveries in modern biology is that the sequence of your DNA is not your absolute destiny. There is a secondary layer of information, a sophisticated control system known as epigenetics. This field reveals that between the fixed letters of your genetic code lies a vast network of chemical switches that can turn instructions on or off, dial their intensity up or down, and fundamentally change how your body functions without ever altering a single letter of the underlying DNA. Imagine your genome as a massive library of millions of books. While every cell in your body contains the exact same library, a skin cell doesn't need to know how to produce stomach acid, and a heart cell doesn't need to know how to grow hair. Epigenetics is the system of bookmarks and highlighters that tells each cell which chapters to read and which to ignore. These chemical tags, such as methyl groups, attach to specific sites on the DNA molecule, acting like physical blockades that prevent the cellular machinery from reading a gene. Other tags interact with the histones, those spool-like proteins we mentioned earlier, to either pack the DNA into tight, silent bundles or loosen it into open, active loops. What makes this system so profound is its responsiveness. Unlike the DNA sequence itself, which remains relatively stable throughout your life, your epigenetic profile is dynamic. It reacts to the world around you. The food you eat, the stress you endure, the quality of the air you breathe, and even your social interactions can trigger chemical changes that flip these genetic switches. This is how nature and nurture dance together. An organism can respond to a sudden environmental shift, like a famine or a heat wave, by adjusting its gene activity in real time. These modifications allow for a level of rapid adaptation that the slow process of evolution could never match. Furthermore, these epigenetic marks can sometimes be surprisingly persistent. While many are temporary, some can remain stable across cell divisions, ensuring that when a cell replicates, its identity and its specific set of active switches are passed on to its daughters. There is even emerging evidence that some of these marks might be carried across generations, allowing the experiences of an ancestor to leave a subtle molecular imprint on the biology of their descendants. This doesn't rewrite the DNA, but it tunes it, preparing the next generation for the world their parents inhabited. This layer of control turns the genome from a static blueprint into a living, breathing logic system. It ensures that the right instructions are available at the precisely right moment, allowing for the incredible specialization seen in complex life. We are beginning to understand that we are not just the sum of our genes, but the sum of how those genes are expressed. By managing the flow of information with such incredible subtlety, epigenetics provides the flexibility and resilience that allows life to thrive in an ever-changing world. It is a reminder that while our genetic code provides the foundation, our environment and our choices help conduct the masterpiece. To truly appreciate the wonder of DNA, we must move beyond the idea of it as a mere storage device and see it as a bustling, high-tech factory floor. Inside the crowded, liquid interior of a cell, DNA doesn't just sit in the dark. It is the center of a frenetic world of motion, powered by molecular motors that are among the most efficient engines in existence. These tiny structures glide along the spiraling strands of genetic material, reading the code with a speed and accuracy that seems almost impossible. They are the physical link between the silent instructions of the genome and the active machinery of a living body. Imagine a specialized molecular engine, a protein complex that latches onto the DNA strand. Using small bursts of chemical energy, it pushes itself forward, step by step, never losing its place. As it travels, it acts like a high-speed scanner, unzipping the double helix just enough to expose the chemical symbols hidden inside. This motor doesn't just read, it interprets. It assembles a temporary mirror image copy of the sequence, creating a mobile transcript that can be sent out into the cell to guide the construction of proteins. These motors must navigate a landscape filled with obstacles, twists in the molecule, tight knots of protective proteins, and shifting loops of genetic material. Yet, they perform their tasks with such endurance that they can copy thousands of letters every minute without a single mechanical failure. There are different types of these biological engines for different tasks. Some are the long-haul truckers of the cell, responsible for copying the entire 6 billion-letter genome during cell division. Others are specialized readers that focus on short, specific passages to guide the cell's daily survival, telling it when to breathe, when to move, or how to fight off an invading pathogen. Their accuracy is maintained by a system of internal checks and balances. If a motor encounters a roadblock or an error in the code, it can pause, call for repair enzymes, or even reverse its direction to ensure the message is correct. But the complexity doesn't end with reading. The genome itself can take on unusual shapes that act like physical signals for these motors. While the double helix is the standard form, DNA can also twist into tight supercoils to store tension or fold into strange four-stranded blocks in regions where symbols repeat. These shapes act like molecular road signs. A tight knot might tell a motor to slow down or wait for a specific signal before proceeding, ensuring that the cell's manufacturing process remains perfectly timed. Without these chemical engines, the information in our DNA would be as useless as a book in a language no one can speak. Their constant activity is the reason you can think, heal, and grow. It is a world of incredible mechanical coordination where physics and biology become one. Every time you blink or take a breath, billions of these microscopic motors are racing along your DNA, turning a silent archive of symbols into the vibrant, breathing reality of your life. They are the unsung heroes of biology, the tireless workers who ensure that the blueprint of life is not just a plan, but a living performance. In the traditional view of biology, the genome was seen as a fixed, immutable blueprint, a set of instructions that remained exactly where they were placed at the moment of conception. However, as we have peered deeper into the molecular landscape, we have discovered a far more restless reality. Your DNA is not a stagnant map. It is a dynamic territory filled with traveling sequences, known as transposons or jumping genes. These segments of genetic material possess the extraordinary ability to uproot themselves from one location and settle into another. They are the nomads of the genome, and their movement has been one of the most powerful drivers of biological innovation throughout the history of life. Imagine a section of code that refuses to stay put. Using specialized enzymes that act like molecular cut and paste or copy and paste tools, these segments can leap across chromosomes, inserting themselves into regions they have never visited before. When they land, they don't just sit there quietly. Their arrival can fundamentally change how the neighborhood works. They might act as a new switch, suddenly activating a gene that has been silent for millions of years. Or they might disrupt an existing instruction, forcing the cell to find a new way to perform an ancient task. While this movement might sound chaotic or even risky, it is actually a vital source of genetic variety. It allows life to shuffle the deck, creating new combinations of traits that simple copying errors could never produce. This internal reshuffling is part of a larger story of interconnectedness. We often think of our DNA as a private heritage, but the microscopic world is much more communal. In the realm of microorganisms, species routinely trade DNA as if they were exchanging business cards. Bacteria can extend small physical connections to pass segments of their code directly to their neighbors. They can even pick up stray fragments of DNA left in the environment by deceased organisms and weave those foreign instructions into their own genomes. This horizontal gene transfer allows populations to acquire new abilities, like the ability to digest a new food source or resist a toxin at lightning speed, bypassing the slow crawl of traditional inheritance. Even more surprising is the fact that DNA carries the echoes of ancient migrations that define the very story of humanity. Deep within your genome are segments that act as molecular footprints, tracing the journeys of your ancestors across vanished land bridges, through ice-covered valleys, and across vast oceans. By comparing these patterns, scientists can reconstruct the history of human movement long before written records existed. Some regions of your code reflect adaptations to the specific climates your ancestors survived. Whether it was the intense ultraviolet light of the tropics or the bone-chilling cold of the north, we are a patchwork of survival. Your DNA is a mosaic made of ancient travelers, viral hitchhikers, and mobile sequences that have been jumping through the genomes of our ancestors since the dawn of life. This roaming behavior turns the genome into a living archive of exploration. It shows that the history of life is not just a straight line, but a complex web of shared information, sudden leaps, and ancient memories that continue to shape who we are today. We are not just carrying a code. We are carrying a legacy of movement and adaptation that spans the entire globe and the entirety of time. As we venture deeper into the molecular heart of the genome, we begin to leave behind the familiar world of classical chemistry and enter a realm where the rules of the very small begin to take over. DNA is not just a biological molecule. It is a physical entity that operates at the breathtaking boundary of quantum physics. Within the elegant twist of the double helix, small physical forces gather, shift, and release in patterns that scientists are only just beginning to decode. It is a world where the mechanical and the informational become one, and where the tension of a molecule can determine the fate of a species. One of the most intriguing aspects of DNA is how it manages energy. To a physicist, the genetic strand is like a tiny, sophisticated spring. When the molecule twists, bends, or supercoils to fit into the nucleus, it stores mechanical energy within its bonds. This isn't just a byproduct of packing, it is a vital part of the control system. This stored tension influences how the molecule behaves when enzymes approach it. Some regions stay tightly wound, sealing off their information like a locked vault, while others are primed with energy, ready to spring open and release their instructions the moment they are triggered. Even a slight change in the physical shape of the strand can alter the distribution of this tension, acting as a structural signal that tells the cell how to react to its environment. But the complexity goes even deeper, reaching down to the behavior of subatomic particles. Researchers in the emerging field of quantum biology have found that the movement of electrons within the DNA molecule may follow quantum patterns. These electrons shift positions between atoms in mere fractions of a second, helping to stabilize the double helix and guiding the formation and breaking of chemical bonds during the processes of reading and copying. Some propose that these quantum effects allow DNA to possess an almost supernatural sensitivity, enabling it to detect subtle chemical changes or manage the high-speed transfer of energy during replication with near-perfect efficiency. While we are still exploring these frontiers, it suggests that the genome is not a passive script, but a responsive, intelligent material that uses the fundamental laws of physics to manage its own stability. This physical responsiveness allows for a level of coordination that seems almost impossible. In every cell, the DNA is constantly vibrating, shifting, and interacting with the water molecules that surround it. Every curve of the spiral attracts a specific arrangement of water that clings to its surface, creating shifting layers of hydration that influence the molecule's flexibility. This molecular dance with water helps proteins recognize the exact regions they need to bind to. It is a reminder that DNA does not exist in isolation. It is part of a delicate, rhythmic ecosystem where chemistry, physics, and hydration combine to create the conditions for life. The deeper we look, the more we see that the genome is a master of structural diversity. It can form knots, tangles, and even crystalline lattices to protect itself from radiation or extreme pressure. It is a material capable of sophisticated mechanical organization, acting as both a biological archive and a responsive engine. We are realizing that the information of life is not just found in the sequence of symbols, but in the very way the molecule is stretched, folded, and energized. It is a quantum symphony playing out in the dark, quiet center of your cells, ensuring that the ancient fire of life continues to burn with incredible precision and grace. As we reach the conclusion of this journey into the molecular heart of existence, we find ourselves standing before a mystery that is both ancient and ever new. DNA is far more than a biological molecule. It is the ultimate record of our planet's resilience. It is the silent, spiraling thread that binds every living thing, from the smallest microbe in a lightless cave to the tallest redwood reaching for the sun, into a single, magnificent tapestry of history. We have seen how it stores data with a density that shames our greatest machines, how it heals itself with the precision of a master surgeon, and how it dances on the very edge of quantum physics to maintain the stability of life itself. Consider the journey this molecule has taken to reach you. Within your cells are segments of code that have remained virtually unchanged since the dawn of life. These are the conserved regions, the essential instructions for breathing, dividing, and processing energy that have been passed through the fire of millions of years without losing a single beat. They are the molecular anchors that link you to the first simple cells that appeared in Earth's primeval oceans billions of years ago. To carry these sequences is to carry a piece of the beginning of the world. You are a living museum, an archive of every victory, every adaptation, and every survival strategy that has ever succeeded in the long history of evolution. But DNA is also a master of the future. It is a generative system, constantly exploring new possibilities through rare mutational sparks and structural rearrangements. It is the architect of the orphan genes, those mysterious instructions that seem to emerge from nowhere, giving a species a unique tool or a new way to sense the world. It is the engine that allows a bacterium to rewrite its defense system in real time, and the clock that measures out the moments of a developing embryo with breathtaking timing. It proves that life is not a finished product, but an ongoing performance, a script that is being rewritten and refined in every cell, in every second of every day. As you drift toward the end of this journey, let the realization settle that you are part of a profound continuity. Your DNA is a map of connection, revealing that even when we are separated by oceans, mountain ranges, or vast spans of time, we are linked by a shared chemical logic. It is the quiet architect behind the growth of the forests, the rhythm of the tides, and the complex architecture of your own mind. It is a world where information is not just cold data, but a vibrant, pulsing legacy of light and shadow, memory and hope. If you feel your eyelids growing heavy, allow the grand story of life on Earth to wrap around you gently and guide you into calm. Don't forget to subscribe to listen to more of our bedtime stories. Close your eyes, take a deep breath, and slowly drift into sleep, into silence, into the boundless, timeless current where all life intertwines. Wishing you a quiet